Good evening, everybody. Welcome to the kickoff of the UCI School of Medicine Dean's Distinguished Lecture Series for the 2024-2025 academic year. I'm Dr. Michael Stamus, for anybody who hasn't met me yet, Dean of the School of Medicine here at UCI. The Dean's Distinguished Lecture Series, or DDLS as we call it, was really designed back in 2019 to bring together influential leaders from healthcare, academia to share their insights, their experience, and their stories with our faculty, staff, students, and the community at large. And while we had a bit of a transitional period because of some pandemic that occurred, we're back at it full strength now, and we're going to have three phenomenal speakers lined up over the next 7 months. And you'll see some cards there, and we'll make some other announcements later, but just so you can understand what's upcoming. But before we get to that, we have a fantastic event tonight. But the program for the DDLS is really around three separate focus areas. And one is innovations in basic sciences. One is speaking to advances in clinical medicine. And like our speaker this evening, one is representing developments in really clinical and translational research and how it really, that sort of bridges the gap between that basic science work that develops the ideas and then the clinical care that results in really revolutionizes the care of patients, especially in the case of tonight's lecture. So tonight I'm honored to welcome world-renowned molecular biologist in the field of angiogenesis, Dr. Napoleone Ferrara, who I know you will love. I had a great pleasure of getting to know him today. He's going to share with us his insight, his experience, and his vision in his presentation titled Preventing Vision Loss Due to Age-Related Macular Degeneration, Progress and Challenges. We'll ask that you hold your questions till the end of the talk, and we'll have microphones available so everybody can participate in the Q&A. Before we get started, please be aware that this lecture is being recorded, and the full recording will be available to you and others on the website in the coming weeks. So without much further ado, I'd like to invite Dr. Daniela Boda, who's our Vice Dean for Clinical Research and Director of the UCI Alpha Stem Cell Clinic, up here with me to formally introduce tonight's speaker. Thank you.
Thank you, Dean Stamos, and welcome everybody to our Dean Distinguished Lecture Series for 2024-2025. Today, it is my absolutely great pleasure to introduce an amazing, brilliant scientist and a wonderful person. Dr. Ferrara will be sharing with us tonight his presentation titled Preventing Vision Loss Due to Age-Related Macular Degeneration, Progress and Challenges. It does promise to be quite a fascinating talk. Dr. Ferrara earned his medical degree in 1981 from University of Catania Medical School in Italy. After completing his postdoctoral research at UC San Francisco, he joined Genentech in 1988. It is there where he spent nearly 25 years working on the isolation, molecular cloning, and biological characterization of VEGF-A, which resulted in the development of bevacizumab, the first anti-angiogenic agent to be approved by the FDA for cancer therapy. It is very rare that you get as a physician to meet the person that has developed the medication that you probably prescribe most on your career. I had this true opportunity, and I think it's extremely inspiring for all of us trying to develop new treatments and cures that somebody of this stature has come to talk to us tonight. But when developing one drug, it's the goal, the holy grail for many of us. Napo hasn't stopped there. In short order, he also has developed the ranibizumab, which is an FDA-approved treatment for multiple intraocular neovascular disorders. In December 2012, he decided to leave the dark side and joined the UC San Diego School of Medicine as a distinguished professor of pathology and a distinguished adjunct professor of ophthalmology and pharmacology. In 2020, he was selected as the Ben and Wanda Hilliard Chair for Disease of the Eye. Dr. Ferrara's research continued to focus on the biology of angiogenesis and the identification of its regulators. According to Google Scholar, we still use Google, Dr. Ferrara has authored more than 300 scientific publications, which were cited more than 130,000 times. He's also the recipient of too numerous to mention scientific awards. Dr. Ferrara has been elected a member of the National Academy of Science since 2006 and has become an elected member of National Academy of Medicine in 2015. It is my absolutely distinct honor to welcome to the podium Dr. Napoleone Ferrara.
Well, thank you very much for both, the Dean and Daniela for this very generous introduction. I think it's a great honor for me to be here. So this is a wonderful place, actually, and I would like to share the work that I've done, actually, for a long time, actually. And we're talking about the blood vessels, so let me point out, for those who are not familiar with this area, actually, the blood vessels, actually, are the most vital, kind of tools, the conduit, blood, and regulatory substance. And then, of course, we all understand the different size, different type of blood vessels, like depending on the size, arterioles, venules, button. But clearly, the most dynamic component of the system is the smaller vessel, the capillaries, because that's where the action takes place, where the exchange can take place between the blood and the tissues. And that's where the growth, the new vessels started from the smaller vessels. And there has been lots of interest, in other, from a developmental biology point of view. And actually, these are events which take place mostly during the embryonic life. There is this concept of vasculogenesis and angiogenesis. Vasculogenesis means the insight or differentiation of cells, from the local mesenchyma. Actually, they form, the cells, they form this primary capillary plexus, which you can differentiate further. And then the more mature type of blood vessel growth is called angiogenesis, which happens also in the adult. There is the sprouting, which is, of course, is very critical. The reason why, of course, the developmental biology aspect has been extremely important. But what is inspiring even more is perhaps, the application, the role of it in disease, for example, in cancer. This is a study published in 1939, extremely imaginative science. They use, what at that time was a state-of-art, tool, which is a transparent chamber that you can put, implant, in a rabbit ear. And they implant the tumor, and they noticed that after a week there was this. I don't know if there is a pointer, but it doesn't. That's probably not. Doesn't matter. Or maybe this. Oh yeah, maybe. No. Well, it doesn't matter. You can see there is in the middle of this growth, there is that's the tumor, and the blood vessels go radially toward the tumor. Actually, and this insightful investigator suggested that the tumor must be producing, they call it blood vessel growth stimulating factor, which turned out to be, of course, a very insightful idea. But this was in 1939, so the tools to advance this field simply did not exist. But this was a very seminal idea. And then there is another really very important, extremely influential work, actually reflected work by Dr. Judah Folkman, which based on this idea suggests that the angiogenesis is critical for tumor growth. So he was probably the first one to speculate the blocking angiogenesis could be a practical way to treat disease like tumors. And then there is another seminal study published in 1948 by Dr. Michelson, Isaac Michelson, just based solely on morphological observation on patients with diabetic retinopathy. He speculated that the ischemic retina produces a factor, an angiogenic factor, which was called factor X, which is secreted by the retina, reaching the vitreous, even the aqueous, resulting in all the problems with the patient with the diabetic retinopathy. This was this seminal idea. Starting maybe in the 80s, there was lots of interest in identifying this, either the tumor blood vessel growth stimulating factor or this factor X, because the idea was that targeting of this molecule, perhaps you could have treated some diseases. Actually, when I started working in this field, which was actually in the mid-80s, there was already quite a few molecules which have been isolated, identified. For example, the most promising was a member of the so-called basic and acidic FGF, which are very potent molecules, probably the most potent angiogenic factor. But there was something very surprising about this molecule. They were not secrete protein. which is very counterintuitive because angiogenesis is a phenomenon which probably requires diffusion in the microenvironment. And this molecule were not very well suited to that. And actually, early attempt to block this molecule didn't have a significant effect on angiogenesis. Even later on, when it was possible to do some gene knockout studies, there was a very minimal consequence of inactivating this factor. So this molecule was potent when applied as pharmacology, but endogenous was very unclear what they did. And so there is this quote in my Dr. Mike Langspoon that says, even though there are a number of angiogenic factors, but it's unclear which one plays a role in angiogenesis. That's what I was very fortunate to give a contribution to this field. Actually, this work I started when I was a postdoctoral fellow at UCSF maybe in 1983, 84. I was working on cells, the pituitary, which are really, even to endocrinologists, are pretty obscure. They're called follicular stellate cells. Cells which don't produce any of the typical hormone like a growth hormone, prolactin, etc. But they have an organization. Actually, I wish I could show you a slide, but in the interest of time. They look like a glial-like cells which infiltrate in between the pituitary cells. And also very often they end up in proximity with the blood vessels. And so there was the possibility that these cells could play a role in the organization of the pituitary vasculature, which is extremely abundant. And actually, in this study, I was able to initiate at UCSF, and then this work was done actually at Genentech. We were able to identify a molecule, an activity which we call vascular endothelial growth factor. And then we had, at that time, the technology is totally different from today. It was extremely time-consuming work. You have to purify a protein, to have a single protein, a single species. And determine the amino acid sequence was the way to go from the protein to the DNA. It is essentially what we did. We were able to to isolate some clone of this of vascular endothelial growth factor. And now, decades later, this was 1989, so 35 years ago, we now understand a lot more about this system. It's very well mapped. There is some receptor called, a class of receptor which is called tyrosine kinases, which is a very well-known classes actually. VEGF receptor 1, VEGF receptor 2, and there is a third receptor. What is very striking that this family of receptors regulated not only blood angiogenesis, but also lymphatic angiogenesis. But this is done mostly by VEGF receptor 3. And this is very complicated on a slide, but essentially the receptor which mediates a signal is VEGF receptor 2, with one which transmits this mitogenic and angiogenic signal. Instead, the VEGF receptor 1 was the first one to be identified by our group in collaboration with a group at the UCSF. Still remaining very enigmatic, but now there is very compelling evidence that's very important in a number of diseases, including disease of pregnancy, like preeclampsia. This molecule does not really signal, but bind the VEGF, prevent from interacting with the signaling receptor. So kind of like a negative regulator of this pathway. So this is probably one of the, believe it, most significant experiment about elucidated the role of VEGF in physiology. This is the knockout of the VEGF gene in mice. What is very striking is that even inactivating only one of the two allele of VEGF, because there are two alleles for each gene. And usually, inactivating one copy rarely resulted in any significant consequence because the other copy compensates. But not in this case. You can see this is a mouse embryo amid gestation. This is on the left side. You have the wild type with both alleles. Looks very nice, blood vessels. And then in the heterozygous, with only, you can see there is hardly any blood vessels in the yolk sac. And at this stage, the embryo either severely underdeveloped is already dead. So this is one of very few, very rare example of a gene which is so critical that if you do a partial inactivation, you have this embryonic lethality. So it is already suggested this was a very important gene, actually. This was published in 1996. And of course, I don't mean to imply that VEGF is the only gene, the only molecule involved in angiogenesis. Actually, this slide is illustrative how there is this look like a symphony, event which need to be so tightly coordinated. It starts from early progenitors in hemangioblasts, which differentiate only in the blood and endothelial lineage. And there is a series of receptors. Actually, FLIC1 is the name of the mouse, VEGF receptor 2. So this is clearly plays a very important role. And there is another pathway that actually, for example, result in the differentiation between the veins and arteries. a family called ephrins, which also tyrosine kinase. There is other factor. But to make a long story short, we have a correct, as an assembly of the vessel wall, you have also these mural cells, which are called pericyte, which play a very important role in disease, like in the diabetic retinopathy. There is a loss of pericyte. And so these endothelial, the blood vessels, they are no longer functional, actually. They become very leaky. And once again, the idea that, angiogenesis required for tumor growth is a very old idea. But at that time, when we started, we published this data, was in 1993. This was literally the first experiment, to try to block, VEGF. This was done 3 years before the knockout. So at that time, we didn't have any genetic evidence. And so we made the monoclonal antibody. And it tested, this antibody in immunodeficient mind, which is implanted with different tumor cell lines. At that time, the expectation is that, you need to block, many factors to inhibit tumor growth. This was the initial idea. So the expectation at that time was very low. Maybe you can see a very small effect. Instead, to our surprise, there was a really very profound inhibitory effect in this tumor model. Like the A673 is a rhabdomyosarcoma, a tumor derived from the skeletal mass. There is like over 90% inhibition. And the same is in like a G55 is a glioblastoma, SK11S1 is a leiomyosarcoma. So a broad spectrum of tumor seems to be inhibited by this system. And this is an experiment called intravital microscopy, where you can image the vasculature in a tumor, actually. And you can see this on the left, actually one of the tumor, the A673. I'll show you the vascular. You can see very tortuous. Actually, already at day 3 there is implanting the tumor. At day 14 there is an extreme level of disorganization. And instead, on the right is the group treated with anti-VEGF antibody. There is a very dramatic difference. The vessel looks much less tortuous, much more normal. Even though, of course, it is small tumors. But the idea was at that time that maybe this could become a therapeutic, so we had this monoclonal antibody, it's a very nice monoclonal antibody. But you cannot use a mouse monoclonal antibody in humans because it's going to be very immunogenic. And so at that time, the technology of that period was humanization, which is a technology, actually Nobel Prize winning technology and essentially looks very conceptually is very simple, but technology at that time was extremely complicated. In other words, you needed to create an antibody which is only the minimum level of mouse residues, and so less than 10%, and the rest is a human antibody. So hopefully it's not going to induce an immune response in human, essentially. This red, this little red, dots, are the so-called, complementary determining region, which are required to bind the antigen. And in reality, it was much more complicated because of some residues, which actually outside, this CDRs, which are very critical of binding the antigen. So this work, actually, the humanization was done by a group, especially I want to take credit Dr. Lem Prester, which was an outstanding expert in this technology. An antibody which we created, which is called bevacizumab or Avastin, essentially reflect the same binding characteristic of the original antibody. Actually, it's about only 7% of murine, does not induce an immune response, and also some species-specific characteristic, only interact with the human and maybe rabbit VEGF, but not mouse VEGF. So it can block in this model only the tumor-derived human VEGF. And this antibody actually started clinical trial in 1997, and the first positive result came in 2003. This paper was published in 2004. Patient with a previous untreated metastatic colorectal cancer. It was in combination of bevacizumab with the chemotherapy. At the time, it was the IFL regimen, irinotecan, 5-FU, and leucovorin. It was very surprising that this was a period, there was a profound skepticism about anti-angiogenesis. There is very negative data, sometimes very controversial data. So at that time, it was a great surprise that this drug, had the benefit in patients. There was a survival benefit, may not seem huge in a few months, but this is extremely difficult to increase, patient survival in patients with advanced cancer. Still, after 20 years, in the survival in patients, mostly measured in months and not in years. Even with immunotherapy, there is some subset of patients which have a prolonged, durable response, but most of them don't have any benefit. I can say that today, this was approved in 2020. Even today, 20 years later, according to the criteria of ESMO and ASCO, which is this entity of, this is still recommended one of the standard of care in colorectal cancer. So this drug had the life, actually. This was a first line, a second line colon cancer. It also became actually very successful therapeutic, actually. As of 2019, before biosimilar emerged, it was one of the major blockbusters, actually. And it was in the top, 10, not the cancer drug, all drugs, actually. It was with Roche, Avastin, actually Roche had the three drugs, Herceptin, Avastin, and Rituxan as three major blockbusters, And now there is a series of biosimilar, which actually probably made, lower the cost, of the drug, which is probably good for patients. And actually, very recently, there's been evidence there is some very exciting combination, for example, with the anti-angiogenic agent with immunotherapy. This is studied in patients with hepatocellular carcinoma, with the liver cancer, actually. And the immunotherapy, neither the immunotherapy nor anti-VEGF alone increase survival. But the combination with increasing survival, this has become now a standard of care. So we're all very excited about that. But what about the main focus of today's talk is what happened in DAI. DAI actually has been an extremely rewarding kind of system to test the anti-VEGF agent. Actually, this illustrated, this is a very classical kind of slide, illustrated the vision in patients with diabetic retinopathy or age-related macular degeneration. A patient with the proliferative diabetic retinopathy, the peripheral vision is severely compromised, but the central vision very often is preserved. So these people can still, in many cases, read, drive, etc. Instead, what is terrible is AMD, because while the peripheral vision is relatively preserved, it's the central vision which was the most seriously compromised. And actually, and actually, the study published in 1994, another study published decades ago in collaboration with a group at Harvard, Josley Diabetes, were able to measure using assay which will be developed in our group the level of VEGF in the aqueous and vitreous in the eye fluid of patient with different diseases like diabetic retinopathy, retinal vein occlusion, etc. And there was almost a perfect correlation between the level of VEGF, and presence of proliferation, actually. And once the disease became quiescent, then the level of VEGF became almost normalized. And this, of course, it was a purely correlative study, but I think it pointed in the right direction. Everybody at that time there was this animal model, which is a monkey model of retinal vein occlusion induced by laser. So there is actually ischemia, which is a powerful stimulus for the release of VEGF. And there is actually, consistent with early study by Michael Sodin in 1948, there is actually this rubeosis of iris. There is the iris becomes extremely red, because there is a proliferation of blood vessels, because the VEGF is produced by the retina and travels across, the vitreous and reach the iris. And this is on the left, is the control with a control antibody. On the right is with the murine antibody, which is fully cross-reactive with the monkey VEGF. There is in those early studies there was a very dramatic effect, almost like close to 95% inhibition, which looks very, to our colleague at Genentech, almost look too good to be true. They didn't believe that this could be a very, very good target. And that's what happened. But then actually, there is this other study. This was a study clearly demonstrated the impact of VEGF in the eye. This is in patients with the macular degeneration, with age-related macular degeneration, actually, with a very, very aggressive form of macular degeneration. What was tested in this case was an antibody fragment derived from Avastin, which is affinity maturity. So 5 residue increase the affinity for the antibody by like a 50-fold or so. So even though this is small molecule, much smaller molecule, not bivalent antibody like Avastin, but still it's extremely potent. At that time, the idea was that this could be more diffusible, so it can reach the target. And this is actually the visual acuity in patient treated, injected on a monthly basis, either with ranibizumab, which is at 2 doses, 0.3 or 0.5 milligram. And the control, this was not an inert control. This was in green, the photodynamic therapy at that time was the standard of care. which is a very little effect. You can see there is this little blip, of increasing visual acuity in green, and the line goes back, like it looks like essentially equivalent to an untreated group. And instead, at the time, it was striking that both those group of ranibizumab, there was actually an increase in visual acuity, which actually is there is even a little bit of dose response, even though when this striking response is been in about 35% of patients. In the remaining 30%, there is a stable vision. But this effect is dramatic and translated in this kind of effect. And actually, this could be some of the cell biology change which happen in AMD. There is actually this the VEGF release, and that there is this growth of blood vessels which become very abnormal, They like, pericytes, they develop, so-called microaneurysm, the little outpouch of the vessel wall, we can burst, it can bleed and leak, And the blocking of VEGF not only inhibit further growth of this neovascularization, but essentially makes the blood vessels more normal. The vessels which are mostly abnormal are pruned. So there is an extensive remodeling of the capillaries. And this clearly explains much of the benefit, which essentially consists in reducing leakage and bleeding, which can result in vision loss. And today there is actually a series of anti-VEGF drug which has been approved. Actually, Avastin has not been approved, but it's widely used off-label because the cost is minimal. It's essentially taken from cancer samples, and actually the cost is like $50 for one dose of Avastin. And then the rest, the second is Lucentis, Eylea, aflibercept, which is an antibody, which is a soluble receptor derived from VEGF receptor 1. And then there is this another drug called Beovu, brolucizumab, which is the poster child for how difficult it is to develop a drug. This is a small molecule, it's a single chain FV, which is so very soluble, actually. And so they were able to give a very high dose, And even though the molecule by itself does not have a very long life, but they gave so much they could last very long. And this drug was approved with FDA, and then shortly after it was approved, this incredible story emerged, that maybe 0.5% of patients develop, vasculitis, which result in blind in some patients. It was due to an immune response, So this keeps you on your toes at drug development. And the latest is the Vabysmo, which is an antibody developed by Genentech and Roche, which bispecific antibodies, an antibody with two arms, which block, VEGF and another molecule called angiopoietin-2. The efficacy is very similar to the one of anti-VEGF, but this drug is given a fairly high dose, 6 milligrams. So last, is the latest. Can be given every 3 months, every 4 months. So this is clearly in advance from this point of view. And now we know there is long-term data that 50% of patients treated with actually in maybe not even optimal dose, but they've 20/40 vision, which means you can read, you can drive. About 10% have actually normal vision. And this result would have been impossible in the pre-anti-VEGF era, where most of the patients at this stage will be completely blind. So, and there is some population study. This is a study, for example, conducted in Denmark, which is a small country with a very good health care. There is a single payer. It showed that this plot, the incidence rate of legal blindness due on top of the black is AMD, does not distinguish all kinds of AMD. And below is a different causes. It showed that after the introduction of anti-VEGF in 2006, there is about 50% drop in the incidence rate of legal blindness. But there is another challenge with anti-VEGF therapy. The fact that the compliance of patient is not great. Actually, this is the data not in clinical trial, in the real world. It showed that there is like a continuum in a European study. It's a retrospective analysis of over 2,000 patients treated with ranibizumab. The top line is the UK, and the bottom line, it pains me to say that, is Italy actually. Patient was seen very infrequently. In France is not that much better, actually. Patient was seen so north to south, actually. Patient was seen very infrequently, like some people receive one injection every year or so. Instead, in the UK, it was much more systematic. The first year, patient received on average five injections. The second year, 2.5. You can see there is already a drop, in visual acuity. So this shows how challenging it is, for patient, to comply. And so this led, to the idea that we need to develop a drug which is longer lasting. And Vabysmo is one example. Another one is another device which was developed by Genentech, which is a port, delivery system, a little implant, in the vitreous, requires surgery. We inject, refill, in this case with ranibizumab. It can last, over 6 months, Initially, they have some trouble about, because now they've fixed, the problem is reintroduced in the market. And so this, the anti-VEGF has had a major impact on macular degeneration. But clearly, AMD, as many people know, is a continuum, it's a spectrum of condition. Like, the early AMD, there is so-called, drusen, there is a minor abnormality, in the retina. And then you have intermediate AMD, there is more and more. And then there is the advanced neovascular AMD, which is divided in two types. One is called the non-neovascular, also known as geographic atrophy. And then you have the neovascular form. And now we know that the neovascular form can be very effective treated with anti-VEGF agent. The problem has been since we have anti-VEGF agent, now the geographic atrophy has become the leading cause of blindness due to AMD. And you see this large area of the retina can become atrophic. That's what it's called geographic atrophy. And once it gets closer and closer to the macula, then you have some severe visual loss. And actually, there is lots of genetic studies trying to understand what would be the risk factor for macular degeneration. There is a very extensive study, actually. This study published in 2016, maybe the number could be even greater. There is some genes with some gene variants which are major risk factor for macular degeneration, actually. Probably the best known is a complement factor H, which is an inhibitor of complement. Complement it plays a role in combating infection, etc. So, but when it's not, controlled, can have a very destructive effect. It can result in tissue destruction. There is others, but the one which has been the main target is complement factor H. So there is in homozygous for certain variant, there is like almost tenfold increase in the risk of AMD. And so this seems to be the ideal target, actually, blocking the complement, activation, actually. The first one to test was, I think, Genentech, with the anti-factor D antibody, which inhibit, some of the key regulator alternative pathway of and this drug, the promising phase 2 data, but 2 large phase 3 data was totally negative. This was the big blow to everybody working in the field. But then there is other approach to target C3 and C5, complement, protein. And actually, very recently, there's been approval of 2 drugs, Actually, they have a very modest effect. They have a very small effect. But in a subset of patients, result in a neovascular eye disease. So this is better than nothing. This is kind of a milestone. We have a drug approved, but there is lots of room for improvement. So what can we do? Actually, together with some colleagues, actually, we thought there could be an alternative approach, because it is known that one of the factors that lead to preceded geographic is the dropout of endothelial cells in the choriocapillaris, that extra retina actually. This is the choriocapillaris below, etc. And we precede the death of retinal cells. So is it possible? That's the idea we contemplate. Is it possible if one could prevent, this dropout of endothelial cells? We could, perhaps, ameliorate, the effect. Now, there is studies show that, actually, in very, very elegant study, in the geographic atrophy, there is substantial reduction of the choriocapillaris density, even compared to AMD. So what do you do that? The idea was you think maybe you could give a VEGF itself, as a protective factor. The problem that VEGF induces vascular permeability, induces leakage, so could not be an ideal therapeutic, at least at the beginning. You could cause, this very same thing that you want to avoid. So the idea we should find a factor that perhaps, does not, protect, endothelial cells, but does not induce vascular leakage. How do you do that? This looks like the million-dollar question. But we knew that there is a very important concept in vascular biology, this endothelia diversity. Look at organs like in the liver, in the heart, in the kidney. Endothelial cells are all different, and there is this heterogeneity. Actually, a number of years ago, we described, studying this diversity, a factor which has a selectivity for a certain type of endothelial cells. In that case, the endocrine gland endothelial cells, And you see, this was done when I was at Genentech. This was done by a genomic and proteomic approach. So we thought maybe there could be other example of a factor which is selective for endothelial cells. How about choroidal endothelial cells? And that's what we try to achieve in our group, actually, to see if we could identify a growth factor specific for choroidal endothelial cells. And actually, to make a long story short, we identify a tumor cell line because the tumor is always the source of everything. It was a glioblastoma cell line, which one of the very few cell lines that don't produce VEGF, don't produce other known angiogenic factor like hepatocyte growth factor. And we thought that this was an interesting, actually, system to study. We use an approach you use in the 80s, purify the protein, actually. The difference, you don't need to do so. The technology is much more advanced, so it requires much less work. And so we're able, actually, to enrich, to partially purify some fraction. And we're able to give an identity to this molecule. Now, to be surprised in a molecule, leukemia inhibitory factor, which is actually is a molecule with a very complex effect. Actually, it's been characterized in some setting as an inhibitor of angiogenesis. But in this case, it was a mitogenic factor. Is a member of the IL-6 family, which is a very important family of regulatory molecules. So this is illustrating this pathway, the LIF, interacting with the LIF receptor, which in turn, interacting with the molecule GP130, which is one of the, which result in a signal cascade, which in a very unique signal cascade. And we're able to demonstrate in our study that actually LIF, in fact, stimulate endothelial angiogenesis when injected in the mouse retina, actually. And we use the state-of-art tools like OCTA angiography to document that. But what is very interesting, actually, LIF works in a model of geographic atrophy. There is a very poor model, but it's a model based on the administration of sodium iodate, which is a strong oxidating agent. And there is actually this is the untreated group in the middle with the sodium iodate. You see the retina it becomes so flat. There is loss of photoreceptor. And instead, the group are treated with LIF. There is actually it's not perfect, but it's a much great improvement in the morphology of this system, actually. But what was very interesting, actually, because there is lots of controversy with the LIF receptor. Today, there's a beautiful single-cell transcriptomic, this omics that everybody's talking about, where you can identify where the receptor are expressed. There is a data set, described by a group at the University of Iowa, Robert Mullins, probably the best, there is all eye tissue. And they were able to inquire for the expression of the LIF receptor as compared to LIF, IL-6 receptor, and KDR is the name for the VEGF receptor 2, which should be the quintessentially endothelial specific receptor. You can see the subunit, which is the IL-6-ST, which is the common subunit, is expressed everywhere, as you expected. And look at the LIF receptor. It's expressed at an extremely high level in the endothelial cells, almost to the same level as the VEGF receptor. There is expression a little bit in fibroblasts in pericyte. But what is interesting, the IL-6 receptor, which is kind of, IL-6 is the typical mediator of inflammation. The receptor for LIF and for the IL-6 is also, as you expect, expressed in monocyte, macrophage, in inflammatory cells. Instead, the LIF receptor is not expressed in these cells. So we thought that this could be potentially very, very useful system, actually, And actually, this is what I was working hypothesis this is the healthy macula, and then in GA there is the progressive dropout of choriocapillaris. And the idea is that if you protect the choriocapillary, prevent preserve the choriocapillaris, you can have a beneficial effect. You try to develop actually with some colleague at Venture. Actually, we try to make some LIF analogs, because LIF has a pretty short of life. So we try to develop some tools that could be potentially useful. Maybe perhaps in not too distant future we can have some clinical data in that. But, I'd like to show a couple of slides, because this is supposed to be a general talk. There is an enormous progress in the treatment of visual loss. There is a new therapy like a gene therapy. Stem cells as yet have not done a great result, but there is lots of interest. And then, of course, gene editing like a CRISPR, and other technologies, optogenetics, which essentially allow to, even with the connection with the retina severe, you can essentially transfect, particular genes, which allow you to, which respond to the light, so essentially bypass the need of a photoreceptor, which is a very important, actually, advancement. This is the gene therapy is already a reality. Actually, for example, there is a drug which is called Luxturna, which was approved by the FDA a few years ago, which is an adeno-associated virus, which is kind of permanent expression. Actually, this was extremely elegant work done by Jim Bennett, and a colleague, in Boston, actually, and there is a mutation in the RPE65, which is an enzyme which is critical for the regeneration of the visual pigment. It is mutated in a number of diseases, retinitis pigmentosa. It's a very complex and genetic, and actually the delivery is not a miracle therapy, but provided it's done early enough, can have a very significant benefit. And now there is multiple drugs have been tested. The challenge is there is a lot of different, conditions, different mutations. And actually, pharmaceutical companies are not interested in treating, a disease which affect, a handful of patients. Actually, distant relative is a kind of, retinitis pigmentosa. They will struggle to find, a therapy which so far has been unsuccessful finding that. But there is a lot to do in this field, actually, in this area. And actually, I would like to conclude here. I want to thank many colleagues and collaborators which have done an outstanding work. I don't include my colleague at Genentech who did too long ago. I spent 25 years there, but there has been it's been a great experience. My colleague at UC San Diego, and also some colleagues like Dan Schwartz, Marco Zarbin, and others have been we've been talking a lot about developing a LIF based treatment. With this, I'd like to thank you very much for your attention.
Thanks for a great talk.
Thank you.
So you indicated stem cells haven't been very successful, but why is that? Because the immune system, the eye is an immune privileged organ,
well, there has been
it seems like a great approach.
Yeah, there have been multiple attempts. The immunogenicity is a formidable challenge, actually. Yamanaka group, they did a handful of study with the patient-derived, iPS cells, and this is an impossible task, because the costs are immense, But they did a study, it was published actually in the New England Journal of Medicine with the Takahashi and colleagues, and the effect was very, very, very small, I think. I don't think it was published in 2017 in the New England Journal of Medicine. But to my knowledge, not progressive. But I know the NEI, the National Institute, actually is conducting a clinical trial now on, I think, iPS cells, But the problem is that unless they are patient-derived, the patient need to be an immunosuppressive therapy for a lifetime, which is an extremely unpleasant thing.
So thank you. That was really exciting. I was intrigued by the large number of genes that you've implicated in reviewing different studies implicated in these eye diseases. And we hear a lot about cancer panels looking for predisposition genes. And I'm wondering what the status of panels is for eye diseases, particularly because they tend to be degenerative. So you think if you could capture and institute some nutritional or environmental changes, you might be able to capture that early on.
Well, these are genetic disorders. I don't think it's the environmental change is probably important, for example, in macular degeneration. But this disease, let me see if I can go back to that. This all unfortunately, most of them are genetic disease, actually, like, some of the different kind of inherited recessive or even dominant, etc. So these are the very tricky one, actually. So, and the only thing that probably could work at this point, I think, is gene therapy, because you can correct, the genetic defect, I'm not so sure that environmental changes could have a great effect. This is kind of the challenge, the frontier now, actually.
Thank you, Doctor, for your excellent presentation. Could you, I'm not a scientist, so I can't talk at your level exactly, but what kind of work is being done to restore vision after you've had severe geographic atrophy or macular degeneration to the point of not being able to read or things like that?
Well, I mean, there is a different. It depends on the stage. You mean you mean the stage where you call legal blindness? That's what you're referring to?
Yes, and I was just wondering if there was any way to redirect existing receptors in your eyes to the parts of the optic nerve that hit the brain. Yeah.
No, there is some study, like one, for example, optogenetics. One approach like that. There is some light response to some light, and you can actually see something, even if complete severe the connection with the photoreceptor. I don't think it's been tested in macular degeneration. It's been tested in more severe, in very severe, genetic condition, where there is a loss of photoreceptors, etc. Macular degeneration, I think, perhaps is not as severe. I think even if in a severe form, you can still see the top, line in a chart, but is it still reduced vision? But there is all this effort that going on right now, and I believe in the next few decades, we'll have a much better idea. Probably it's not work which happen in a couple of years, but we'll have a lot. I'm convinced there will be lots of progress here. And clearly the gene therapy, the pharmacological therapy with anti-VEGF works very well in wet AMD. Dry AMD is very seems to be more difficult to treat. But the course is more indolent, because of wet AMD could lead very rapidly in blindness, in legal blindness. Dry AMD, GA is a much slower course, Well, I mean, I don't know. I don't think so. I think maybe in next decade there has been much more. I mean, I don't say perhaps I misspoke. I think there already is a huge progress. The first gene therapy was approved in 2017, and I'm already in 2024, and a number of other approaches have been approved. So the field is moving very rapidly. There is very reason to be optimistic. I don't know. It's very difficult to tell that, but there is certainly things that looks way better than they look, just a decade ago.
I'd like to take you back to cancer therapy, if I may. You alluded to the seminal studies of Judah Folkman with the the inhibition, essentially, of angiogenesis. And there was tremendous excitement at the time. Yeah. And that excitement didn't last too very long. But then your wonderful study came up with the monoclonal antibody, and that worked tremendously well in colorectal carcinoma. But could you elaborate in the broader sense of that type of therapy for other solid tumors?
Yeah, the work by Judah Folkman was very imaginative, very innovative. But at that time, they didn't fully isolate, the right angiogenic factor. They look at different molecules like endostatin, etc., to not to be a bit, more challenged. But it turned out to be the anti-VEGF is an effect. And I'm not going to claim it's a miracle therapy, because treating solid tumors is extremely difficult, even with immunotherapy, which, as I'm sure you know, is a very exciting therapy. Most of patients don't have this dramatic survival. But what has been very interesting is the combination of anti-VEGF, with immunotherapy, with anti-PD-1 and the PD-L1, has been tested, become a standard of care, like in renal cell cancer, in liver cancer. There is been incredibly, sometimes more than additive benefit between the two. So this, right now, this is the state of art of in the anti-cancer therapy, combination with immunotherapy, etc. So that's what I think is the present, and the future hopefully will be even better, actually, when people find the new targets. Another new target, for example, inhibition of HIF, is a little bit, technical. My HIF, hypoxia-inducible factor, is one of the regulators of, in response to hypoxia, regulating of VEGF and many other molecules, And it was extremely difficult to target because of the structure. Now there is a molecule, actually, an inhibitor of one of the form of HIF, which has been approved with the FDA. Is quite effective in renal cell cancer because there is a 50% of this cancer mutation in the von Hippel-Lindau gene, which is actually one of the so there is an oncogenic activation of this pathway. Probably will not work as well like in macular degeneration because there is no such activation. But this could be certainly a very important area that
Very quickly on that point, with the clear cell carcinoma of the kidney, as you know, that's a HIF. Related thing with tremendous angiogenesis.
Yes.
The combination therapy that you mentioned that's beginning to appear now in various publications, is that working very well for that particular cancer, the clear cell carcinoma?
You mean with
the kidney?
With the HIF inhibitor?
Well, no, not with the HIF inhibitor. Your immunotherapy plus.
Yeah, but I think it's pretty effective.
And has anybody used the HIF?
The HIF has been actually very recently approved as a third-line therapy.
In that
in the renal, in the clear cell.
Yeah, but in combination or not?
I think in the third line is as a monotherapy is compared with the mTOR inhibitors. It's better than mTOR inhibitors. Still, it was first approved in the von Hippel-Lindau syndrome. Actually, it's a genetic condition where there is actually activation of HIF because of mutation of this pathway, resulting in this very highly angiogenic, neoplasm, and this was the most obvious, target. But renal cell carcinomas, 50% of this mutation is progressing. I think the approval is only in third line, but maybe it could be that in time it will do even better. Actually. Thank you.
And our tremendous and wonderful speaker.
Dr. Ferrara, if you please join me up here, I'd like to present you with a gift of our appreciation. It says UCI School of Medicine, Dean's Distinguished Lecturer, Napoleone Ferrara, MD, September 17, 18th, 2024.
This is wonderful.
Thank you so much. Thank you. Let's get a quick picture over here.
Oh, yeah, sure.
I got it. Let's have one, Jonas? How about a great round of applause once again? And I want to thank a few people who helped make this event possible, including Field Applied Innovation, and I don't know if Errol Arkalik is still here or not, but he's our host here, and thank him for the hosting. And all the people behind the scenes in the Dean's office, including Emily Dawidoff, Teresa Waters, Claire Draper, Jessica Banfield, Kevin Wynn, Londa Garcia, Matt Miller, and many others. Thank you also to the crowd this evening. Thank you all for being here and supporting us. And it's really you who have made our Dean's Distinguished Lecture Series a success. So there should be a handout at your seat that announces our next two speakers in 2025. We have Dr. Nancy Carrasco from Vanderbilt coming in January, and then we have Adrian Hernandez coming in April from Duke. So we will continue to bring you great speakers from industry, from academia, who are really changing the world of healthcare and investigatory work. And thank you again for joining us.